Good afternoon, ladies and gentlemen. Welcome to today's webinar. I am Dr. Xiaowen Liu, representing Pharmacology Discovery Services, to share with you our capabilities in vivo pharmacology and the pharmacokinetic services. PDS is part of Eurofins Discovery. We are their partner lab. Eurofins Discovery offers a panel of research capabilities, including chemistry, in vitro assays, safety efficacy, ADME toxicology, and the in vivo efficacy models. We can do in vivo PK services and the efficacy models at PDS. We are offering in vivo pharmacology expertise. We provide services in non-GLP safety, pharmacokinetics, efficacy studies in rodents. PDS offers more than 300 validated models that are ready to test your compound of interest. We are very collaborative and flexible to assist clients from study design to data delivery. We have a lot of experience and skills covering a broad range of different therapeutic areas to provide animal model to test the efficacy of the compound. And we routinely provide custom in vivo models to meet specific project needs. Our facility is located in Taipei, Taiwan. We are ALAC accredited Bavarian lab. Our operation is in compliance with Office of Laboratory Welfare Assurance. That is critical to win opportunity from projects supported by US government grants. And our animal procedures are also in compliance with European Animal Welfare Standard, as well as Korean standards. The company operates in compliance with ISO 9001 certification. We have a facility larger than 15 meters square and with more than 50 years of CRO experiences. We are known formally as Penn Labs. Currently, we have 40 employees. The average tenure is more than 10 years. We own biosafety lab, level 2 labs. That's why we can perform effective animal models. Currently, we have 27 animal holding runs and 19 procedures. With more than 300 efficacy, PK, and safety models, we cover antimicrobial models, CNS pen models, gastrointestinal, renal, inflammation, allergy, metabolic, as well as oncology efficacy models. We also can do in vivo PK and safety pharmacology at PDS. PDS has a lot of experience handling different types of test articles, including small molecules, biologics, cellular agents, bacteriophage, vaccines, liposome formulated, and nanoparticles. And we can administrate different dosing options that are common in animal models, including intradermal, intracerebral ventricular, intramuscular, intraperitoneal, intratracheal, intravenous, as well as subcutaneous, topical dosing, and vaginal dosing. We do short and long-term continuous IV infusion, and we always adjust our dosing schedule according to the study protocols. Not only can we handle the in vivo animal studies, we also provide ex vivo analysis. We perform hematology, blood chemistry analysis. We can analyze surface intracellular protein by flow cytometry, and we can run bioanalysis to measure drug concentration in plasma or urine. And we do cytokine analysis using Luminex and protein analysis using ELISA, as well as semi-quantitative protein evaluation using Western blot. We can also measure RNA levels by using RT-PCR. And we outsource the histopathology work to our trusted lab. We have the stem slides reviewed by certified pathologists. Antimicrobial portfolio is our strongest therapeutic area covered at PDS. We have provide in vitro and in vivo services. For the in vitro services, we do minimal inhibitory concentration and MyC assay for more than 800 strains. We have a collection including 400 genomically characterized strains from CDA, CDC, and FDA isolate bank. We have multi-drug resistant clinical isolate. We can also do fungi, molds, and yeast studies. And our procedures meet with industry standard protocols. For antimicrobial characterization assays, we can do resistant emergence, killing kinetics, combination testing, bacterial cell lysis, hemolysis assays. For in vivo models, we have a comprehensive portfolio of more than 100 models with CDC and WHO priority pathogens in immunosuppressed and neutropenic animals. The endpoints are usually being the microbial counts in tissues or infection survival. The following table shows you the pathogens and infection models we have at PDS. I will use the MARSA strain in wound healing model as an example to show you the data, the typical data and protocols we provide to the clients. In a brief summary of the protocols, you can find the strain we use in this model, USA 300, and a brief study procedures, including the animal strain, the gender, the body weight, and the inoculant count, and how we perform the wound puncture, and the treatment length, frequency, and how we do the tissue homogenation, and how we measure the bacteria counts in the brief protocol. In a typical data figure, we provide two endpoints for this study, including the bacteria counts and wound closure. On the figure, in the figure on the right and on the left, we can see the bacteria counts in vehicle-treated animals from day 1 to day 14, and the drug-treated animals, the pathogen counts from day at 26 hours after inoculation to day 14. It shows the mupirosin significantly suppress the infections in this model. And the other endpoints, the wound closure, we can see the antibiotics did not accelerate the wound closure in this model. And please note that at around day 14, the wound closure in this model reached 90%. Therefore, we also offer another more sensitive wound infection model in diabetic mice, which is more clinically relevant. As you can see in the picture here, on day 5, the animals have very severe infection. The wound size become bigger compared to the original puncture. And on day 14, the wound closure reached around 15%. That gives us a larger dynamic window to access if a drug has wound healing effect in such model. And the benefit of this model is this is a common issue in diabetic patients, the slow wound healing processes. So it's a very good model we provide at PDS. And another popular portfolio we offer at PDS is inflammation, allergy, metabolic in vivo models. We cover autoimmune disease, animal models, including arthritis, inflammatory bowel disease, cirrhosis, and lupus. And for dermal inflammation, we have atopic and contact dermatitis. Highlighted in green star here indicates popular models that we perform many times in a year at PDS. The dust mite-induced atopic dermatitis is very popular. And for cirrhosis model, the IMQ-induced models are frequently requested and conducted at PDS. And we also have the delay type hypersensitivity models. For the digestive systems, we offer gastric irritation, the NASH model, and TMBS-induced colitis models. And I know some of the participants today are interested in NASH model, so I will use it as an example to share. And for the respiratory system, we offer allergy and OVA-induced asthma, LPS-induced COPD models, and the bleomycin-induced lung fibrosis model is also very popular model and frequently conducted at PDS. And we also have obesity models induced by high-fat diet. For the renal failure, we have the acute and chronic ischemia reperfusion model. Other models such as Miles assay, that's a VEGF-induced vascular permeability assay conducted in guinea pig. It can be used to test the effect of the anti-VEGF. For the NASH model, this one we use the choline deficient amino acid defined high-fat diet to induce NASH in animals. The endpoints we'll offer for these animals include the serum ALT, AST, and liver body weight ratio. As shown in the graph here, the elevation of ALT and AST indicate liver injury. And at as early as week 2, we can see the diet can induce a significant elevation of these liver enzymes, and administration of positive control, which is a PPAR gamma agonist, pioglitazone, reduce the liver enzyme elevation significantly from week 2 to week 12. And similarly, for liver weight to body weight ratio, the positive control also significantly reduced the increase induced by the diet. And oftentimes, fibrosis needs to be examined by histopathology. Therefore, we also offer histopathology analysis that will be reviewed by certified pathologists in a collaborator's lab. We can stain the section's liver with hemotoxin and eosin to illustrate the tissue structure, and Mason's trichrome MT stain, which stain the collagen. That's indicative of fibrosis processes. As shown on the right analysis figure here with HE stain, we can identify the steatosis by different score and generate a scoring result as evaluated by the pathologist. And we can show the lobular inflammation, hepatocyte, cellular blooming disease state in these animals after treating with the test compound. For the MT stain, we can score the fibrosis, and then overall NASH plus fibrosis score can be provided to evaluate the overall effect of the drug in such model. Similar to other preclinical CROs, we also offer oncology models. The typical models including the human cell line derived xenograft models in immunodeficient mice and the syngenic models with immortalized mouse cancer lines in immunocompetent animals. For example, using the human leukemia cell line HL6 injected subcutaneously to the back of the mice, we can measure the tumor volume over time. With the positive control used in this study, mitomycin, it shows a significant reduction of the tumor size in this model. And what is shown on the right here is a typical overview of our models that can be found in the website and in such link. And in this brief overview, the summary of procedures can be found, and a typical turnaround time is also available. And using the same cell line, we can also do IV injection to create a disseminated disease model, and the endpoint is animal survival. In addition, we can do flow cytometry analysis of the percentage of human CD45 positive cells in this model to evaluate drug efficacy. This year, we offer a new model, MC30I syngenic model. This cell line is very unique. It responds very well to T-cell checkpoint therapies, as shown on the figure here, using the anti-CTLA4, PD-L1, and PD-1 antibodies. The tumor size was reduced significantly compared to untreated animals. And the last animal efficacy models we offer at PDS, but not the least, is the PAN in vivo models. We actually have a very comprehensive PAN models thanks to our experienced technical director, Linda. And we have been offering acute inflammatory and neuropathic PAN models at PDS for many years, including postoperative PAN, formerly induced PAN, the visceral PAN induced by acetic acid, and the PQ. For the neuropathic PANs, we have a nerve ligation model, charm and banana model. Also, we can use the chemotherapy compounds such as Taxol and oxaliplatin to induce the peripheral neuropathy in rats. Alongside, we also have efficacy models such as depression induced by forced swim, or analysis of animals' anxiety state using the elevated water maze, or seizure induced by chemical or electrical shock. We also offer side effect profiling in the locomotor activity, animals coordination, and drug tolerance and withdrawal. We offer the oxaliplatin induced neuropathic PAN this year, and this is a typical result shown in such a model with 6 inducer injection with oxaliplatin in rats. After 17 days, we can induce a hypersensitivity to mechanical stimuli in these animals. And for example, using a well-known painkiller gabapentin in this model, as soon as 2 hours after gabapentin administration, the animals has reduced threshold to mechanical stimuli provided by Wolfram monofilament. And additionally, we can also use a cold allodynia method, applying acetone to assess animals' response to cold sensation. To better support the efficacy studies, we offer PK services at PDS. We can do PK service in mouse, rats, hamster, guinea pig. We can collect whole blood, plasma, biophysis, urine, and tissue homogenate. In a standard PK setup, we can do intravenous oral gavage administration of compounds in 3 animals per time points, total 8 time points. If the test article supply is limited, we can do mini PK using 2 animals per time point, 6 time points totally. And we can help client to identify the solubility of the compounds in the solution and measure the concentration using LC-MS-MX. And oftentimes, clients would like to know the maximum tolerated dose of the compound before they decide on the dosage they want to use in the efficacy study. So we also can do that at PDS. Once the client is satisfied with the PK and the efficacy results, they would like to know some preliminary toxicology study results for their test articles. So we also offer repeat dose toxicology studies for 7 and 14 days at PDS to assess clinical science, hematology, blood chemistry, urine analysis, and histopathologies. We also offer safety pharmacology evaluations, including modified Irwin test, which evaluate animals' neurological, autonomic, and physical states. And we often offer custom assays to clients. For toxicology studies, we have experience in renal tox, and we can also assess GI side effects at PDS. Our clients include US government, small and large biotech, academia researchers, and big pharma. We have been offering services to National Institute for Allergy and Infectious Disease, part of the US NIH supported grant for many years. We are currently in the fourth year contract at the moment. That indicates we provide quality data in compliance with the animal welfare and procedure operation, and we have been able to provide a report format suitable for the government review. If you are interested in our animal models, you can simply go to the website or go to the Google and type in Pharmacology Discovery Services.com. In the search box, you can key in the keyword or model you are interested, and you can find our one-page protocol or simplified information about our animal operation procedures and typical data on your own from the website. If you cannot find those information, please feel free to contact us. We have different sales representatives support clients in different regions. We have Nicholas and Tony support the North America, and Ricardo support Europe, and myself, Shaowen, support all the clients in the APAC region. A unique feature of PDS is that we have very experienced technical directors responsible for their area and support each study. We have Linizo. She is very experienced, has been with the PDS for almost 2 decades, is an expert in infectious disease. And we have Linda supporting the pain and safety studies, and Jimmy Bao responsible for the inflammation, metabolic, GI, and renal animal models. And all 3 of them are based in the US. And Ken Shen and myself are supporting the PK and neurodegenerative migraine model, which are new models currently under development at PDS. Both of us are based in Taiwan. For the operation side, we have Dr. Ken Shen as our lab director, oversee the business and the lab operation in Taiwan side. And we have 4 study directors. Lucy responsible for the antimicrobial, Gwen responsible for CNS pain, PK, and safety. Taiyu is responsible for the gastrointestinal inflammation and the metabolic studies. And Pony is the expert for our oncology studies. And thank you for your attention. I would like to take any questions you may have.